Welcome to the latest edition of our video series, Ask the Experts, where I ask the leading authorities in a wide range of topics related to brain-gut microbiome interactions in health and disease. Several weeks ago, I invited Dr. Beate Ritz, professor at the Departments of Epidemiology, Environmental Health, and Neurology at UCLA, to speak at one of our brain-gut microbiome centers symposia about her seminal research in Parkinson's disease. Even though Parkinson's disease is generally considered a neurological disorder affecting particular areas of the brain, it has become obvious in the last few years of research that the disease can affect different body functions, including the GI tract. In addition, most excitingly, it's becoming clear that the microbes living in our gut play an important role in this devastating disease. It is a pleasure to share today Beate's exciting research into the role of the gut microbiome in the development of Parkinson's disease.
I'm a professor of epidemiology. I'm an MD from my background and actually trained in psychiatry, which in Germany includes neurology. So I have actually a co-appointment here in the neurologic department. And as you can see, I have been pretty much working for more than 20 years now in community-based research, mostly in the Central Valley, in terms of Parkinson's. But as Emeran just said, we are also doing studies on pregnancy exposures and child neurodevelopment. And I'll mention that as well. And all of that is also in the Central Valley. And so the new frontier really is the microbiome. I've been thinking about this for more than 5 years. We started with the pilot study 3 years ago. It took me 4 rounds of applications and we finally have an R01. So it's a long process, right? The R01 is supposed to look at pesticides as well as Parkinson's disease, and both in the context of the microbiome, in a sense that pesticides might actually act and generate neurotoxicity through the microbiome. And why that is so important is because it has now been shown, and everybody knows, you know, Parkinson's is a motor disease. Here are all the motor symptoms. This is how we diagnose Parkinson's. But really, it has been known now for a very long time, there are all these non-motor symptoms, and some of them are really strongly gastrointestinal related. The strongest symptom is constipation. And the idea is that the peripheral autonomous nervous system is also affected. The vagus is affected, and therefore the gut motility is affected. And more recently, it has been shown that alpha-synuclein, which is the protein that agglomerates in the brain, gives you the Lewy body of Parkinson's and leads in the end to the demise of dopamine neurons. They are found in the gastrointestinal tract around the autonomous nervous system endings as well. It has also been shown that alpha-synuclein, which is misfolded and therefore aggregates, that these aggregates can, in a prion-like fashion, get synuclein to misfold and therefore have more and more protein agglomerations in the end. But also these misfolded proteins, if they get, for example, through the vagus to the brain and finally into the dopamine neurons, that could be one axis for all of this to happen. So some Parkinson's might actually start in the gut. And it might have to do with whatever happens in the gut that causes this misfolding of alpha-synuclein. And then the question is, why are these alpha-synuclein proteins then moving up the vagus to the brain? There are some studies in Denmark that showed in patients who had a vagotomy, this didn't happen probably at the same rate because they had a 30% lesser chance of developing Parkinson's. And so that's some of the only human data we have, but in animal models, we have been seeing some of this happening as well. And we now actually have a colleague in neurology who's trying to model a lot of these alpha-synuclein templates and what influences these proteins to then agglomerate. So what we did in our pilot study is we collected stool samples from Parkinson's patients and controls, household controls, as well as community controls. We did see some differences in the gut microbiome, and there's this long list of bacterial genesis and other differences that we identified, but whether these microbial shifts really hold up when we are looking at more patients, we don't know. We split these patients up in duration of disease as well as medications they've been getting. Right, but what I'm actually really excited about is this recent data that a student of mine produced. And what we did here is we used our pesticide data. And just for these 200 people on whom we already have the bacterial information, thanks to Jonathan, you can see that there are two types of pesticides, and this was hypothesis-driven, microbial pesticides and microbiocides that seem to, when you're exposed, these are not patients, these are actually the controls, that seem to actually shift what you're seeing in the gut microbiome, or at least in the stool microbiome that we collected. So there's a first idea of pesticides actually ending up disturbing or changing the microbiome.
What is the OP pesticide again?
That's the organophosphates, sorry. And they don't do anything. So they are kind of the negative control in this case. It's the microbial pesticides and the microbiocides that changed the alpha diversity. When you're looking at differential taxa abundance, you can see that there's also a little bit of a signal for the organophosphate pesticides, but we don't really know what this means. And clearly the microbicides and microbial pesticides also are influencing the taxa abundance. Again, we don't really know what this means yet, but it's a good start to follow up on. And these are all the people who have been working to collect samples and see Parkinson's patients for the last 20 years. And I want to thank them, especially Dr. Bronstein and Dr. Keener and Dr. Bordelon, who've been seeing all these patients. Questions? Oh yeah, and so we don't have stool samples from any autistic children or their mothers in pregnancy, but what I've been hoping is to use the metabolomics approach, because we have serum samples from women in pregnancy who later had a child with autism, that we can maybe do metabolomics analyses and backtrack to possible microbial differences in their gut, and then understand whether there's any influence of the microbiome on whatever happens in pregnancy, which might actually be related to the immune status of the woman more than anything else. I have also participated in a study of 200 women at UCLA that we followed during pregnancy, and we have 3 urine and serum samples and metabolomics on those. And we have also placenta samples, and one interesting question could be to look at the placental microbiome, if it exists, but there's some people thinking it might now exist, and the metabolome that we are analyzing in these same women. So there's lots of really exciting research we can engage in. But of course, the Parkinson's one is the most immediate and the one that I'm really happy we get to do in the next 5 years.